Hello. Today we are talking about DNA structure and components, chromatin structure, and some classic DNA experiments. Let's start off with some of the components of DNA. As you know, DNA has the deoxyribose sugar. Remember, ribose is a 5-carbon sugar. Deoxyribose for DNA means there is just a hydrogen, not an oxygen hydrogen or a hydroxyl group attached to the 2' carbon. Our 3' carbon does still have the hydroxyl group attached, and that becomes very important in DNA replication. It also has a nucleotide, also has a phosphate. And if we can think about the sugar, the deoxyribose sugar is a building block for our nucleotide. We have 5 carbons, 1', 2', 3', 4', and 5'. The phosphate group is attached to the 5' carbon, and the base is attached to the 1' carbon. The 2' carbon is the one that differs between DNA and RNA, since ribose has the hydroxyl group here. And the 3' carbon with this hydroxyl group on it is where the next nucleotide will be added when we talk about DNA replication later in the semester. There are 4 types of nitrogenous bases. 2 of these are purines. They have a double ring structure. In fact, you can see the purine adenine right here and the purine guanine down here. So adenine and guanine are the 2 purines. The pyrimidines have a single ring structure, thymine and cytosine. Now, you don't have to memorize the specific structures, but I do want you to know the full names of these bases, not just A, T, G, and C. And then I want you to know which ones are purines and which ones are pyrimidines. It's helpful if you remember that purines are the double ring structure and pyrimidines are the single ring structure. So let's see, we had thymine and cytosine. Note that these are the 4 bases in DNA. If we had RNA, we would not have the thymine, we would have the base uracil instead. A complex of a sugar and base is a nucleoside, and the complex of the sugar, base, and phosphate is the nucleotide. And we've already talked about how that is constructed. In the lectures, we mentioned that Watson and Crick utilized information from Chargaff's work and from Franklin and Wilkins' work in order to come up with their model of DNA. So let's think about what some of the features are of the Watson-Crick model and how they incorporated information from Chargaff, Franklin, and Wilkins in order to get those features. So we know that DNA is double-stranded. Part of the reason we know that is if you just string the nucleotides along, it's not going to have the coiled configuration or the helical configuration or the correct diameter. And remember, Franklin and Wilkins had the 2 nanometer diameter. And they noticed that in their experiment. So if it's double-stranded, that means we have to have 2 strands, and somehow those 2 strands have to have a consistent width. We can think about that as due to the base pairing so that we always pair a purine and a pyrimidine. So we're pairing a double ring and a single ring structure, keeping that a constant width. So we have complementary base pairs. So that we always pair adenine, across from that will always be thymine, and across from cytosine will always have guanine, and vice versa. Note that there are hydrogen bonds that can form between these base pairs. There are 2 hydrogen bonds on a T base pair and 3 hydrogen bonds on the GC base pair. Those hydrogen bonds are important for strain recognition, but the thing that really holds these 2 strands together are the hydrophobic interactions. Between adjacent base pairs, so between one base pair and the ones above and below it. So that says between BP for base pairs. If we look at the distance between one base pair and the next, that distance is 0.34 nanometers. So 0.34 nanometers between base pairs. And that is something that was another number that Wilkins and Franklin came up with. And one turn of the helix is 3.4 nanometers. This is the third number that Franklin and Wilkins came up with. So they came up with 2 nanometers, 0.34, and 3.4. Chargaff's contribution, remember he did base pairing studies, and he found that the amount of purines was always equal to the amount of pyrimidines. That makes sense if we're always pairing a purine with a pyrimidine. The amount of adenine was always the same as the amount of thymine. Again, A and T would be the 2 that pair. Guanine amounts, same as cytosine amounts, and that makes sense because the G and C pair. There are other features of this. It is a plectonic coil. And that's something that means the strands actually have to be unwound. The hydrogen bonds have to break apart and unwind the strands in order to get the 2 strands to separate. And let's see, alternating major and minor grooves. I don't have something that shows that on this picture, but if you refer to the lecture slides, you should be able to see that as well. So I think that gets some of the components of the Watson and Crick model. So this says circle one nucleotide that's in this molecule of DNA, but not at the end of the molecule of DNA. So that means we have to get the right phosphate with the right sugar. Just remember up here, the phosphate is connected to the 5' carbon of the sugar, and the 5' carbon is the one that's not involved in the ring. So if we look up here, this phosphate must go with that sugar and that base. So that would be one such nucleotide. If we wanted to do that on the opposite strand, this phosphate would have to go with that sugar and that base. There we go. So there are a couple of examples. So another property of Watson and Crick's model was that DNA had a right-handed coil. Now that right and left coiling bit was always something that kind of confused me in organic chemistry. And most forms of DNA do coil right-handed, but there is a structure called zDNA because DNA is dynamic. So at times in the cell, some DNA may coil in a left-handed manner, and that would be zDNA. In order to figure out how this stuff coils, I finally realized that you can look at a circular staircase. Stand at the bottom, put your hand on the railing, this outer rail right there, and then walk up the stairs and turn. The hand that's on the railing is the coiling direction. So if my hand is on this rail right here, that is going to be a right-handed coil. I went around through the kitchen and found some noodles. And if I look at this and do that same sort of walk up the stairway, I see that these noodles are both coiled in a left-handed orientation, whereas these are coiled in a right-handed orientation. And then I found some cool curly-Q noodles. These also coil in a right-handed orientation. I found a piece of string as well. In fact, I actually still have it. If I look at this string is coiling in a right-handed orientation. Now I have a molecule of DNA here, and this is actually coiling in a left-handed orientation. This image shows DNA coiling in a right-handed manner, and this may be a little hard to see. It didn't photocopy super well, but I think you can see the sugar phosphate backbone here and here. This is also coiling in a right-handed manner. Well, like I said, the coiling for DNA is not going to be static. It's not static, it is dynamic. It does change at times. And if you coil DNA more tightly in one place, then it's probably going to be looser in another, and vice versa. Supercoiling occurs when we have extra coils put into the DNA, and typically that's due to uncoiling the DNA in one place, causing supercoiling in a place downstream. So if you think about a telephone cord, I know most of you have probably never seen telephone cords like this, but that curly coil and how it kind of coils around and sometimes coils too much, that would be like a supercoil. So we have both positive supercoiling and negative supercoiling that can occur. There's an enzyme called topoisomerase. This is a class of enzymes that affects supercoiling. The idea is that having too much coil provides tension for the DNA molecule. And so the topoisomerases actually nick the DNA, allow it to uncoil and reseal it to relieve that tension and make that DNA be in a more happy manner in the cell. But let's look at what positive versus negative supercoiling is. Positive supercoiling occurs when DNA is overwound and it twists back on itself. There we go. So I've overwound the DNA and it's twisting back on itself. Let's see if I can do that a little bit more. Oh, there we go. And the positive supercoil is going to be in the opposite direction of the DNA helix. Well, this string is a right-handed coil, and since the helix DNA is also typically right-handed, a positive supercoil will be left-handed. So DNA is a right-handed coil, then you would expect a positive supercoil to be a left-handed coil. Now, negative supercoiling occurs if we under-rotate the DNA. Let's see if I can get that to do. Hold on, I have to. And it will also supercoil. But this supercoil coils in the same direction as the DNA. So DNA typically right-handed, so a negative supercoil will also be in a right-handed manner. And that's what these diagrams here are showing you. I tried to show you with the string here, and then I've got some examples below. Well, so think about normally with a Watson-Crick model of DNA, we have 10 base pairs per turn. And that's under the Watson-Crick model. So think about that as our normal model. So if a DNA molecule is 30 base pairs long and has 20 complete turns, then do you think the molecule is underwound, negative supercoiled, or overwound, positive supercoiled? So 300 base pairs with Watson and Crick, 300 divided by 10 is 30, but I only have 20. So I have under-rotated this DNA, which is negative supercoiling. So here's another one. DNA molecule is 100 base pairs long and has 20 complete turns. Well, let's see. Normally with Watson and Crick, 10 base pairs per turn. 100 divided by 10 would be 10 base pairs, or 10 turns. But wow, I've got 20. I've got twice that. So this is over-rotated. Positive supercoiling is over-rotated. And the other option here is that in its relaxed state, that means we have the expected 10 base pairs per turn. So I've got some examples with my string. So you can see these pictures close up. The string here has a normal right-handed coil. This is my string. The ends of the string were twisted and the supercoil was allowed to form. And for each picture, you want to determine if the string is more tightly or more loosely coiled than its resting state, and if the supercoil is a positive or a negative supercoil. So before we answer that, let's review this one more time. Positive supercoil, over-rotated, and the supercoil is in the opposite direction from the normal coil. Negative supercoiling, under-rotated, and the supercoil is in the same direction as the normal coil. So this is why it's important to be able to determine what is a right or a left-handed coil. So if I look, this is the supercoil right here. If I look at that supercoil is coiling in the right-handed direction. So the supercoil is a right-handed coil. This is meaning that we must have it less tightly coiled than normal, and we have a negative coil. So even though the questions were phrased in a different order, I looked at the direction of the supercoiling first and answered that part first before going to the other parts. Now let's look at figure B. This supercoil is going in a left-handed manner. That's telling me that the string is more tightly coiled, it is overwound, and I have a positive supercoiling going on. And you say, well, when does this happen? One time this happens is during replication. In replication, the strands have to come apart. And that's what I've tried to show here. My string had 3 strands, so I pulled one separate from the others. But that's the same sort of idea. And when I just fix the ends and I uncoil parts of it, I get supercoils formed. Do those supercoils look like they're going in the right or the left-handed manner? These supercoils are going in a left-handed manner, which means I have positive supercoiling going on. I encourage you to walk through this diagram yourself, as well as these examples. Always, you can find a string. If you want a string, stop by my office sometime, and I will be happy to give you a piece of my string and play with it. Yarn also works. Shoelaces might work. I don't know. I haven't tried that. But I encourage you to play with it so that you feel comfortable with the orientation of the regular coiling of the strand plus the supercoiling. Well, let's look at chromatin structure then. So chromatin structure is basically the complex of the DNA and the proteins that make up the chromosome. And we say the name for that complex is chromatin. Now, there are various levels of packaging to make a chromosome, and various proteins are in charge. But what we're going to look at first are the complex of the DNA with the histone proteins. These histones, DNA, by the way, has a negative charge because of all the phosphate groups on the outside part. And the histones have a positive charge, which means it can interact easily with the DNA. The amino acids that allow that positive charge to occur are lysine and arginine. And that helps those proteins to be able to bind electrostatically to the DNA. We have an octamer of 4 different, so 2 of each, histones that come together to form this little yellow part right here. And the DNA wraps about twice around that octamer. That octamer of histones is 2 each of the proteins H2A, H2B, H3, and H4. And the DNA wraps right about twice around there, and it's right about 147 base pairs of DNA. And then you notice that there's a little bit of DNA that's going between these different beads on a string. That DNA is the linker DNA, Linker. And the protein H1, actually think of that H1 as being right here, is the histone that complexes with the linker DNA. So the histone H1 is needed. And that histone is H1 is actually needed to get proper coiling. So when there's no H1, we get these beads on a string, but once we have the H1 present, we do get coiling and then multiple layers of coiling. So it will coil, it will sort of stack, and then those stacks will fold. This first stacking is called the 30 nanometer fiber. And then as that coils more, we end up with a 300 nanometer fiber, and it even coils more and more, particularly as it goes toward a metaphase chromosome to get additional levels of packing, because a whole bunch of DNA has to fit in an itty bitty little space. So let's see, the second level of packaging occurs when the nucleosomes are coiled into a 30 nanometer fiber. That is, you guessed it, 30 nanometers in diameter. And then those loops of the 30 nanometer fiber are actually anchored by a nuclear scaffold that's composed of non-histone proteins. So this nuclear scaffold, I can't think of a blank that would be there. I think nuclear scaffold was supposed to be my blank. And the scaffold is anchored by non-histone proteins. And that helps the fiber to condense into that metaphase chromosome. It's actually kind of cool. I think in the lecture I mentioned this, that if you get rid of the histone proteins and the DNA and look just at what's left of a metaphase chromosome, you have the nuclear scaffold, and it looks like what you would expect the scaffold of a metaphase chromosome would look like. So I thought that was kind of cool. Well, the other thing that we talked about quite a bit in this periods of this time of lecture, the set of lectures, is some of the classical experiments. And certainly we've mentioned some of these. We've mentioned a bit about Watson and Crick, Franklin Wilkins, and Chargaff, and you should go back and look at that in more detail. But we also looked at Griffith, Avery McCarty McLeod, and Hershey and Chase, among others. I encourage you to go back in your notes, and for each of these experiments, go and look up what was the major question they were asking. How did they try to do this experimentally? So this would be like the simple idea of their experiment. Can't spell experiment. There we go. And then what did they observe in their experiment, and what sort of conclusion can they draw? I'm not going to do all of these. They are all answered for you in the key anyway, and you certainly have them in the notes, but I wanted to look at Hershey and Chase. It just happens to be one of my favorites, and it's one that we covered this time. So Hershey and Chase were trying to figure out what was the infectious agent in a bacteriophage. Now, a bacteriophage is pretty simple. Protein coat, nucleic acid. So we've got the DNA hidden inside the protein. And they know that the bacteriophage T2 can infect E. coli and cause lysis of E. coli. So their goal was to determine, was it the protein or was it the DNA that caused the lysis? In order to do that, they had to distinguish between protein and DNA. And they did that by radioactively labeling the bacteriophages. They grew them for multiple generations in S35, which is radioactive sulfur. Now, proteins have sulfur, nucleic acids don't. So this labeled the protein coats of all the newly made bacteriophages. And in a separate flask, they grew the bacteriophages by allowing them to infect E. coli for multiple generations in radioactive P32. Phosphorus, that's in DNA, but not in proteins. So this labeled the DNA, and they used P32 for that label, and this labeled the protein. And they used S35. Then, after they had some labeled bacteriophages, they allowed their labeled bacteriophages, and again, this is two separate flasks, they allowed them to infect unlabeled E. coli. So there's no more radioactive label present. And then they, as soon as infection could occur, but before the bacteriophage were lysed, they sheared off the whatever would be outside of the bacteria by putting them in a blender, and separated the proteins from the cells, or separated the cells from the culture media. And then they looked to see where was the radioactivity. The S35 radioactivity was in the fluid around the cells, and the P32 radioactivity was in the cells themselves. Now, in both of these cases, they could take some of the cells and allow them to grow a little longer, and sure enough, they would lyse. So in both cases, we got infection. However, only P32 was into the cells, which means that P32 labeled DNA. So DNA was the infectious agent in the bacteriophages. I encourage you to look at all of these experiments that were in this chart, as well as the other experiments that were mentioned in the lecture slides, and fill out this type of information for each of the experiments. And the key does have this completed online. Thank you for your attention to this lecture. I hope it was helpful. Please ask if you have any questions. Have a wonderful day.